All right, so yeah, this is our lab for ENE480. It's focusing on indoor air quality analysis, focusing on the ventilation of buildings in MSU's campus and how they relate to the transmission of infectious diseases. So we're going to start with the importance of ventilation here. Proper ventilation of indoor spaces is very pivotal in kind of mitigating diseases, but it is also crucial in kind of increasing student success. There have been certain points made about the presence of CO2 in a room and therefore kind of the lack of ventilation there, bogging down the brain a bit and worsening student performance. And these things can be seen to kind of increase as the occupants of a space increases. And so, you know, ideally the ventilation rate increases in a room as the occupants increases kind of with that. To be acceptable in terms of ventilation rate, either the room has to achieve a rate of 15 cubic feet per minute or maintain a concentration of CO2 inside that room below, you know, within the range of 700 parts per million higher than the outside air. In kind of taking these measurements of CO2, we can develop a further knowledge base on the presence and behavior of infectious diseases such as COVID-19, which have very kind of similar particle sizes and decay rates, as we will kind of talk more going forward.
Moving on, we discuss how the ventilation relates to transmission of COVID-19. According to the CDC, COVID-19 is often spread through contact with infected respiratory fluids through inhalation of airborne droplets or particles and respiratory droplets coming in contact with mucous membranes of the body through direct contact with droplets or indirect contact by touching surfaces on the face. However, adequate ventilation in a room would remove these infected droplets or particles, limiting transmission to others in the room by long-range transmission, which adds on to the previous slide of why ventilation is very important. However, it is also still very strongly recommended by the CDC to continue to wear a mask, as these really help limit the amount of infectious particles that normally leave a sick person through their mouth, through a cough or a sneeze, that could infect through short-range transmission or by the particles building up in a poorly ventilated area.
So what was our study? We used a CO2 monitor in various different locations to determine the relationship between CO2 levels and the ventilation in the room, as well as determine whether they had appropriate ventilation or not. These rooms included an off-campus home kitchen, a research lab in the PBL building, numerous rooms in the engineering building, a computer lab, a research lab, and also an off-campus apartment. The aim of the entire project was to utilize these different rooms with different ventilation systems and see how the data varies for each. Is there enough ventilation in this room to appropriately combat the CO2 levels students produce through their rebreathing during class time? These are some of the questions that we answer in this project.
All right, so to begin this lab, we as a team determined where we wanted to take samples. It was mostly to see the differences between, you know, somewhere with really great ventilation and somewhere with, you know, not so great. So, you know, the research labs would obviously have pretty good ventilation, and maybe something like a computer lab with a lot of students, you know, wouldn't. And when picking these rooms, we had to choose them where there wouldn't be a lot of, you know, non-human CO2 emissions, because that would throw off our readings. So, you know, like open flames and such. And once we collected that data, we downloaded it off the monitor, put that into Excel, and you know, using it as a spreadsheet, we were able to manipulate the data in ways to, you know, make graphs and analyze our readings. And then part of those readings was using the outside CO2 rate, CO2 reading, it's included in one of the equations. And for all those, they were lower, below 450 parts per million, which is a pretty good reading. All right, our first sample was generously provided to us by our professor, Dr. Maston, in the form of her home kitchen. Uh, pretty average kitchen, family members coming in and out cooking. So, uh, in analyzing that, uh, so I guess this is a great time to explain, what we did with the data. So, out of all the readings, we found the maximum CO2 reading in parts per million, and then took all the data points after that, and then plugged them into an equation, which we can provide you at a later date if you're interested. But using that, we were able to find the time since the maximum, and then using the equation, we found the decay rate of CO2 in the room. Here specifically, it was 0.184 per hour, which is pretty average rating. So the CO2 decay rate is basically how CO2 leaves the room. And then we also, using that data, found the rebreathed fraction, which is how much CO2 you're breathing in that someone already exhaled. And the lower the better in most cases. And here is actually 0.0026, so pretty good reading. All right, our second sample was taken in the forest pathology lab in the plant biology building, room 34 specifically. There's usually like a max of 9 people in that room, but it's usually around 4. It's a rather large room, like, I don't know, 40 by 30, and it has a lot of ventilation in the form of, filtered ducts and fume hoods and such. So, we picked that as a way to see, what is the CO2 rate in a really well ventilated room with a few amount of people. And with that, we found, the CO2 decay rate was 1.62 per hour, which is pretty great. And the rebreathe fraction was very, very low, which is 0.00086.
So our third sample was also taken, was taken in the engineering building on MSU's campus. It was on 3rd floor, taken in the graduate level computer lab. In this room, 2 exits are connected to the, there are 2 exits, and it's also connected to the main ventilation system. It is open all day, but really the only spike in occupancy of this room was kind of between 10:15 and 11:30 in the morning there, most likely for a class project of some sort. And as it kind of says here, the maximum occupancy of this room is around 35 people. However, this kind of peak group was somewhere between 15 and 20 students. To kind of baseline this analysis, the outdoor CO2 was measured kind of early in the morning. It was especially low at that 401 parts per million. And coming back inside to the lab, I went ahead and placed that monitor on the back corner on top of a bookshelf. This was kind of to avoid any nearby vents or open windows or anything like that, or any, obviously, indoor carbon sources which weren't in that room, but also away from students directly breathing on the monitor to kind of avoid those dramatic spikes. The maximum level was measured to be 674 parts per million, and that was for that time frame of 9 a.m. to 1:15 p.m. The average rebreathe fraction was measured to be 0.00254, so quite low there. And the decay rate was quite slow. It was that 0.0829 hours to the -1.
So for the 4th sample that we used, I did the monitoring in room 2250 of the engineering building. It's a lecture hall with only 1 computer for professor use, around 40 chairs for students. However, I monitored this a little bit before my class time and a little bit after my class time, just to see the way that CO2 reacted after students came into the room as well as left the room. There was only about 20 students in the room, with 1 leaving halfway through. The CO2 levels within the room increased gradually as the lecture hall filled before class and during class. We see a peak around the end of class in that graph at the top right. And I believe this to be when we see an increase starting around 40 minutes into the class period. And this is because there's more people in the room rebreathing CO2, and the CO2 levels increase. And around the 160-minute mark of me monitoring, everybody began to get up. And what we do at the end of this class is we have conversations between our groups. So I believe that during this group time, with more people communicating, more people talking, more breathing, rebreathing CO2, we have a peak at the 170-minute mark on that top right graph. The second graph on the bottom shows us the decay rate of CO2 over a 35-minute interval after the class had ended. You can see a steady incline in the CO2 decay rate over time. Based on this graph, we inferred that as students leave the room, the rebreathed CO2 levels not only go down, but the decay rate of carbon increases over time.
Lastly, we have our fifth sample, which was taken in room 1001 at the 1855 place. So room 1001 is basically, it's just a meeting room that's at the first floor of the building. This room has one exit, one entrance, same way, and is connected to the common lobby area. And it's often used for meetings or training, such as initiate at state. We noticed that the room can hold a maximum capacity of 31 people, where 30 people and then the one person being the presenter. However, the time we witnessed when we took our reading, we noticed that 23 people were there after the monitor was placed for the day. The outdoor CO2 that we measured came out to be about 400 ppm, part per million. However, the monitor was placed at the far right corner of the room, as mentioned previously, to avoid any complications of such matter. Finally, the maximum level of CO2 that was measured when we analyzed our data came out to be 43 ppm. And when we use the required equations, we get to find that the average rebreathed fraction was measured to be 0.00134, and the decay rate of the sample turned out to be 0.0447 hours in burst. And right here we have the results in a summary form, which in a table that's easier to view. And please take a second to take some time if you want to go through the data and have any adjustments.
So in conclusion, we found that COVID-19, well, we know that COVID-19 has similarities to CO2 in particle size, decay rate, and also movement throughout a room. CO2 monitoring in this way can help us model the risk factors of COVID-19 in different environments. The behavior of a COVID-19 particle operates similarly to that of CO2, so the metrics such as concentration and rebreathed fraction and rates can give us insight into COVID-19 exposure and also help us in improving our ventilation systems and improving our policies in terms of mask wearing. It should be noted that mask usage can also be a double-edged sword of sorts. It helps to protect against the spread of COVID-19, but it can also increase personal rebreathed CO2 as you rebreathe that CO2 that you directly pump out right into the mask. It was found in a study by the University of California, Berkeley, that increased consumption or inhalation of CO2 decreases student performance at a high rate. There are also health implications that we found. Clean air promotes a strong state of mind and ability to accomplish goals. And with high levels of CO2, students and regular people alike may not have the best tools to accomplish their goals. And that's why air quality is important. The air quality that we monitored in all of these sites was nowhere near bad. There could be some improvements like increased ventilation. It should be noted that it's winter and the heating systems are on. So there could be differences if this was tested in the summer when the AC might be going. Increased ventilation in classrooms like those in laboratories would improve the air quality and lower the risk of spreading diseases and viruses like COVID-19. We found some ways to improve the data collection process. We could have had better predictions of class time and monitored throughout more than one class. It's notes for the future. And instead of specific time measurements, we could have done 4 full day monitorings instead of specific time hours in the day. And another point is we placed our initial monitor with a sticky note saying, please don't move this. And somebody did move it, so that kind of pushed back our data just a little bit, but we ended up getting everything together on time. So we'd like to thank you for listening, and if you have any questions, please comment them below. Thanks.